Good afternoon, and thank you for joining our webinar today. My name is Jon Rowley. I'm the founder and chief product officer at RoosterBio. And today's webinar is going to be on radically simplifying the production of human mesenchymal stromal cell extracellular vesicles that have the potential for both rapid clinical translation and scale up. I am both heavily dependent upon and eternally grateful to the extended RoosterBio team who has really generated the majority of the content that is within this presentation. So at RoosterBio, our goal is to radically simplify MSC production. And since the cell culture is really the main bottleneck within extracellular vesicle production, we are also radically simplifying MSC-EV production with our plug-and-play MSC expansion and extracellular vesicle collection systems. So how do we simplify MSC production? We do this in three different ways. First, we make it really easy to establish MSCs within new programs. You no longer have to have 5 to 10 years of experience with MSCs. Anybody with basic cell culture skills can now generate significant quantities of MSCs from which to collect extracellular vesicles from. And we do this in a few different ways. We have extremely high quality cell vials. We have BioProcess media, and then simple protocols that anybody can follow. We have MSCs from multiple tissue sources, both bone marrow, adipose, as well as umbilical cord. And then we have kitted these up into EV collection systems so that, again, anybody can very rapidly get to generating very large volumes of extracellular vesicles for their research applications. Now, once these projects have really evolved and you're ready to actually enter something like product or manufacturing process development, much of this happens within industry, but we have made it easy to scale up the research into product development activities. So we do this by having manufacturing friendly product formats with established and robust protocols. And then we've demonstrated comparability between traditional flask-based expansion as well as a scalable bioreactor expansion, of which we have gone up to 50 liters in scale. And then third, once these product and process development programs are ready for the clinic and you're ready to treat patients, we have CGMP versions of both our cell banks as well as our media available so that you can rapidly start clinical production of your extracellular vesicles. Again, we have essentially off-the-shelf working cell banks with all of the regulatory documentation. We have master files on file with the FDA for both the cells and the media, and we have multiple companies that have been using our products throughout product and manufacturing process development and are currently treating patients today. For those of you who are not familiar with mesenchymal stem stromal cells, they are the foundational technology for multiple cell and gene-based regenerative therapeutics, right? So they're used really across the spectrum in hundreds of clinical trials every year with tens and tens of different indications ranging from regenerative medicine applications to immunomodulatory applications such as graft versus host disease or Crohn's disease. The cells are also used as the basic building block for many engineered tissues and organs. And because of their extensive safety profile, they've been used in tens of thousands of patients with a great safety profile. They're used in many cell-based gene therapies. Now, what we'll be talking about today is really the MSC-derived materials, which is the secretome, and specifically the extracellular vesicle aspect of this. And right now, there's hundreds of companies that are developing products that include MSCs. Many of them are our customers, and they all need the same thing with it, which is an industrialized supply chain so that they can very rapidly get through product development and into clinical production. So the MSC-EVs have similar regenerative capabilities that the native cells have, right? So there's lots of publications out there, just a few listed here, ranging from orthopedics to cardiovascular all the way through wound repair, that have demonstrated that the extracellular vesicles that are secreted from MSCs have many of the same regenerative capacities. And this is why MSC extracellular vesicles are so popular. And one of the few position papers that the International Society for Extracellular Vesicles has is based on specifically MSC-EVs. Most recently, they've been used in several COVID applications. There's lots of clinical trials right now going on with MSC-EVs for COVID. And why EVs are better than living cells, and this is still the jury's out, but they do make cell therapy easier, because your product is no longer a living, breathing cell that you have to keep alive. It is a secreted molecule, complicated molecule, but it's still secreted, and has much better cold chain logistics, better stability, and more straightforward engineering. And then there's definitely a safety aspect where you're not giving a living cell to a person, but rather a non-living entity. So that's why I think MSC-EVs have been so popular as of late, because of the therapeutic capabilities built in, as well as some benefits over cell therapy. So at RoosterBio, many of us are manufacturing gearheads through and through, right? And so when we think about extracellular vesicles, we think about the production process, and then we try to, again, make this easy for the end user. So we kind of break the manufacturing process down into 3 different pieces: the upstream process, the downstream process, and then the analytical component, and where each of these then subcomponents are broken down into individual unit operations. But for the upstream process, right? So first you have the cell culture. This is typically the most expensive, the most time consuming. Again, this can take several weeks to grow up enough cells from which you need to generate the extracellular vesicles from. And so, what you'll see in orange is that over time, your cells will go up. And once you've plateaued, that is when, and you've hit your maximum number of cells that you're going to get, that is when you want to start extracellular vesicle collection. Now, there are some people that will collect the extracellular vesicles throughout cell expansion. We believe that this leads to inconsistencies within the extracellular vesicle populations, even more than you already have. And so just to standardize this piece of the process, we believe that it's going to be best to first grow up the cells and then have a separate EV collection stage. After you grow the cells up, you generate the EVs and then you collect them. And again, we have products specifically for that. And your extracellular vesicles in blue those numbers will go up, and you will have a heterogeneous population of EVs if you characterize them. As you take them into downstream processing, which is essentially the concentration, separation, and purification of the extracellular vesicles from the cell culture media, you can get to more of a homogeneous population. It's still heterogeneous, but it's not as heterogeneous as your raw populations. And from these go into the analytics, where you look at both the safety as well as the quality aspects, the quality control. And then from this work is you get your CQAs, the critical quality attributes, and this is the data the FDA wants that to know that about your products before you take them into humans. Again, at RoosterBio, we focus exclusively on the upstream process. Again, we make it easy for people to grow up the MSCs, harvest the extracellular vesicles to take into downstream processing. There's lots of different kinds of downstream process technologies, and so I would say that there's a lot of variation on what people do, and so we're trying to specifically standardize the upstream process. And the technology platforms that we have focused on is really all around the manufacturing of the cells, and then how you collect the extracellular vesicles that are generated from the cells that are cultured. And what you'll find with MSC manufacturing is that as the platform evolves and scales up, it does drive the economics of MSC supply. So what you have here, I'm going to spend a little bit of time on this graph, is your lot size. How many cells are you needing to generate? This is in billions, and this is on a log scale. This is not linear. And then you have these S-curves, or they're typically called the technology S-curves in the innovation literature. And this does go across platforms, but they dictate that every technology platform is good for about a log or two of productivity, at which time it maxes out, it gets harder and harder to achieve higher yields, and so you need to move to the next technology platform. And so then on the left is your production platforms: T-flasks, multi-layer vessels, and then three-dimensional bioreactors. And then this helps you get from tens to hundreds of millions of cells that you're able to achieve in T-flasks, billions of cells to the low tens of billions that you can get in multi-layer vessels. But really, true commercially relevant lot sizes are north of 100 billion cells. This is only possible to be generated within suspension bioreactors, single-use bioreactors that are often used in the monoclonal antibody space that lots of people, including RoosterBio, has adapted to MSC culture. And then as you do scale up, the economics is impacted. The main metric that we follow is the cost per million MSCs. As we think about extracellular vesicles, where you need billions of cells, you could think about the cost per billion MSCs. So multiply all of these numbers by 1,000. And if you need to be spending tens of thousands of dollars per billion cells, then your EVs are going to be really, really expensive. And really the main way to get your costs into something that's going to generate a good business model for you is in suspension bioreactors, where your lot sizes are north of 100 million. However, if you get into these bioreactors at smaller scale, start your clinical production, and then you scale from then. You don't have to do your phase 1 clinical trials at a trillion cell lot size. But what we do is we help people get into these systems very quickly, both in the 10-layer flask as well as the bioreactors, demonstrate the comparability, and help them very quickly get into clinical production. So the working hypothesis that we have at RoosterBio is that the extracellular vesicle field is a need of tools that simplify generating significant cell volumes from scalable manufacturing processes with standardized particle collection with a rapid path towards clinical translation. And we do this a few different ways, right? So we simplify MSC production. I like to say, we built into all of our process isn't just good quality control, but it's quality control that matters. All product lots must expand to by 100 to 150 fold in a 5 to 7 day time frame. And we also help people get to these large cell numbers by providing much larger cell volumes. All the other cell products that are on the market today that you can buy are 500,000 to 1 million cells. We have a scaled down 1 million cell version for small scale studies, but we also have 10 million as well as 100 million cells that are at the same low population doubling level, so that people can get from 100 million to 1 billion to tens of billions of cells per manufacturing production lot. We also have good bioprocess media that is paired with our cells that helps them get to at least 100 million cells within the first 2 weeks, or first week, usually within 4 to 5 days. And then if you passage it once, you'll be north of 1 billion cells at a fraction of the cost and a fraction of the time of all the other systems that are on the market. The leading serum-free system and the leading serum-based system. You barely start out with 1 million cells, and you can see that even after 2 weeks, you're barely at 10 million cells. So trying to generate enough EVs for an in vivo experiment can take several weeks, cost tens of thousands of dollars, and this has really been slowing the field down. If you actually do an economic analysis of, again, the top serum-free MSC system, where you have the cells and the serum-free based paired media, as well as then your traditional MSCs, it can cost you $40,000 to $70,000 to generate 1 billion cells from which to collect extracellular vesicles from. In our systems, you can do so for less than $8,000, and it's not just the cost. This will take 40 to 50 days to generate 1 billion cells, where we can generate 1 billion cells with our 10 million cell vials in less than 10 days. And this is, again, a fraction of the cost. So that cost per billion cells is just dramatically lower, and most of that is driven through good media design, good quality control, and high volume cells at low population doubling levels. So good cell banking strategy. And we do this through productivity metrics, right? So the productivity of your cell culture media is really, really key. And what that is, it's defined in the Regenerative Medicine Glossary as millions of cells produced per liter of media. So with all of these really expensive systems, in plasma-based culture, you're talking about around 20 million cells per liter of media, which is fine if you're doing cell experiments in 24-well plates. You don't need that many cells, but when you need billions of cells, you need to improve that. And so so our media was designed with productivity in mind. We have a batch culture. You don't have to do very expensive media exchanges. And so again, you can get to almost 200 million cells produced per liter of media. This is with our bone marrow cells. Our cord MSCs are even greater than this. And what this does is drive tremendous efficiency into EV production. And we do this without sacrificing quality. We have the most comprehensive certificate of analysis on the market, where we not only look at your standard ISCT analytics, such as the flow cytometry and multilineage differentiation. We also have the growth potential, as well as potency-related aspects, such as the inducible immunomodulatory capability of the cells, as well as looking at a half a dozen angiogenic cytokines secreted on a picogram per cell per day basis. And we also have this available over multiple donors. Right. So if you're going to go into the clinic, you have to really understand what donors give you good EVs, which donors give you bad EVs, or are all donors good for your specific indication? As it relates to EVs or for MSCs, the way I like to think about it is the best donor that gives you the best cartilage production in MSCs may not be the best one that gives you the best immunomodulatory capabilities. And so it's really important to learn that, and you can't do that without access to multiple donors. And we have donors over multiple tissues available for purchase at any time. And then what we've done is kitted up an EV production system that consists of our cell vials, our BioProcess media, both available in what we call development grade for product development experiments, as well as our CGMP grade that's branded under our Clinique Control brand name. And these help you get to the large cell volumes. And then we also have an EV collection media that is, I believe, the only media available that's actually QC'd for low particulate quality control, which is then specifically for extracellular vesicle collection media. Again, we have master files on the file for our CGMP versions of our products. And there are multiple companies that are taking these into clinical development as we speak and treating patients. So what we do then is we also provide standardized protocols to our customers for the bulk MSC expansion. What people do with the bulk MSCs, whether they're bioprinting an intravertebral disc, whether they're making a cell therapy, whether they're collecting the extracellular vesicles, or whether they're gene modifying those cells, they're all doing something different. But we help them with that bulk cell expansion, where we go from our cell vial, whether it's 1 million, 10 million, or hundreds of millions of cells, that are all made the same way. This is what the consistent CMC across scales mean. They all have the same population doubling level. They all have the same cell isolation, the same population doubling levels as they go through the cell baking process. And then we have the recommended P3 and P4 expansions to get cell yields from hundreds of millions to billions to tens of billions. These are, we have a recommended process for getting to multi-layer vessels for both R&D and clinical production, as well as for bioreactors, right? Whether, again, from tens of millions to tens of billions, depending on the number of cells that you're starting with, across lots of different scales. And we help people, get in there, generate the cells, generate the extracellular vesicles. And what we've done is we've already done the comparability between the 2-dimensional and 3-dimensional cultures over multiple donors. We're publishing this right now, and we have really good comparability of the function of the MSCs across the multilineage differentiation, the traditional ISCT cell surface markers that you would expect, as well as the functional aspects, the multilineage, sorry, the immunomodulatory capabilities of the cells, as well as the angiogenic cytokine secretion profile. And then from the extracellular vesicles, we've been characterizing these in both in 2 dimensions and 3 dimensions, as you can see here. They look very, very consistent. And one thing that we found is that extracellular vesicle production is higher in 3-dimensional bioreactors for a couple of different reasons. One, cells do grow up to higher densities in bioreactors per liter of media, so this drives particle count per mil of media. However, and other people have also shown this, is that in this dynamic 3-dimensional suspension bioreactor environment, you do get more EVs per cell. When you look at the critical quality attributes of these extracellular vesicles, they do have all of the small RNA that you would expect to find in the extracellular vesicles, as well as the protein content from all the typical players, including the tetraspanins that are bound to the membrane. And we've also done some bioactivity to show that these do have the typical bioactivity. So this is the second to last slide. So we really believe that robust extracellular vesicle production starts with high productivity MSC cell cultures. And we really have designed these systems for maximum productivity that give multi-log enhancements in EV production while maintaining EV quality. So the leading competitor MSC systems, it can take you a good 20 days even to get to 100 million cells. And if you start out with our 10 million cell vial and one bottle of media, you can be to 100 million cells in less than 5 days at a fraction of the time and cost, with a 28% increase in extracellular vesicle yield. If you take this to 1 billion cells, you can get over 200 times the EVs in a fraction of the time at a fraction of the cost. It's 200 more experiments, or just much larger experiments, because everybody is always extracellular vesicle EV limited, right, in doing their experiments. And then as you get into bioreactors, we give the people the ability to do 3 logs higher, 1,000-fold increase in EV yield by moving into these systems, which then this is what is enabling the rapid clinical translation of extracellular vesicles with these systems. So we really believe that that we're having an impact on the industry. We're helping people very rapidly get into product development, very rapidly get into clinical translation, interact with the FDA, with our master files, and really pave a path to accelerate regenerative medicine. So if you're interested in learning about these, please come to our website. Click on our extracellular vesicle production systems. Leave us a message. Send me an email. Send anyone at RoosterBio an email, and we would be happy to talk through how we can get your lab up and running with MSC-EVs quicker than ever before possible. So thank you very much for your time. And again, if you have any questions, just send us an email. Thank you very much.